Good afternoon, everyone. It is a great honor and pleasure for me to represent Yikon Genomics to share some thoughts and useful information about the non-invasive PGT. So whether it is a time for us to move forward to the non-invasive PGT. So I hope this talk will bring you through and have further discussion afterwards. So this is a slide showing the development of the PGT. First, it was a cell-based, biopsy-based method. Conventionally, that approach requires a sampling from one or two blastoma at the cliff stage or 5 to 10 trophectoderm cells at the blastocyst stage. So given the fact that the purportance of the embryo is well recognized, but there's still quite a few potential issues for performing this cell-based assay. So as you can see here, the key thing is that this cell biopsy method may damage the embryo and will have a direct impact on the embryo development. And also the long-term effect has still not clear about how much and to what extent this biopsy will do to the embryo and future development. And also the biopsy that requires a very highly skilled embryologist, as well as specialized equipment and training and practice. So this is also a risk of there's only a few cells being sampled, and that could be lost during the transferring from the dish to the testing tube. So given the above all potential issues, so there's a lot of research and tries to look for alternative approaches. So with less invasiveness and also easy to perform. And that is bring us to the stage of the non-invasive PGT. So the idea was from the NIPT actually, because this NIPT is a sampling of fetal cell-free DNA being released into the maternal blood, which enable the detection of very diluted proportion of embryonic genetic materials for chromosomal abnormalities. By the same principle and mechanism, the embryonic materials can also be released from the apoptotic cells into those blastocyst fluids, as well as the cultured media. So during the in vitro culture in a dish. And research has shown that the presence of embryonic genomic DNA in the blastocyst fluid, the real-time PCR has been used to amplify and monitor a few multi-copy genes in the paper published in 2013. And they further applied with whole genome amplification plus with the microarray to prove the principle of the possibility of using the BF as the alternative source for PGT. And also, similarly, the embryonic genomic DNA can also be found in the span culture media, and that has been reported by many researchers since 2013. And there's several attempts was made to utilize the culture media as a source for the embryonic DNA and used for the pre-implantation genetic testing or diagnosis, such as for the X-linked disorders, alpha thalassemia, etc. And so it laid down the foundation of this new generation approach for the PGT, which is either the non-invasive PGT use the span culture media or the minimal invasive using the blastocyst fluid. as a source for the embryonic genomic DNA. And is that the non-invasiveness or minimized invasiveness would help. The span culture media is treated as the clinical waste actually after the transplantation. And also releasing the blastocyst fluid is also part of the standard embryonic vitrification procedure. So therefore, collecting these SCMs or BF does not have a additional impact on the embryo, as well as the invasiveness of and it is not as invasiveness as the PGT using the T biopsy as an example. So this slide shows you the standard workflow of our NICSI assay, the non-invasive capacity screening assay. So on the top of the flow, you can see the work is being performed in the embryology laboratory. The ICSI is currently required for NICSI application, and it is mainly used to control or minimize the source of maternal or even paternal contaminations. And the granule cells were largely removed before the ICSI. And after the injection, you were culturing your embryo until day 3. And then when the media change takes place as the routine procedure. And also, we introduce an extra step of removing any residue of the granule cells where can be introducing any maternal contamination in a later stage. And the spent culture media is then subsequently being harvested on the day 5 or day 3, whenever the embryo is ready for vitrification. So then the collected culture media is being passed to the genetic lab for the downstream procedures of NICSI, where the whole genome amplification is being performed with the Malbag technology, and the library is being prepared for the next generation sequencing. So usually it would take 1 to 2 million roll reads would be sufficient for assaying each of the samples. The sequencing data from the NGS platform will be directly transferred to our analysis platform, which is called ChromeGo. And ChromeGo is having equipped with the AI-based automated analysis and for the cell embryo ploidy and also chromosome copy number variations. And then the test reports will be produced automatically after the analysis for the clinicians and geneticists to review. And this slide showing the major advantages of NICSI techniques. And it listed as below. The first of all is in non-invasiveness, obviously, because the embryo is remain untouched, and we only collect culture media for the assay. And the second, there is literally fairly easy procedure. All you need to do is collecting the culture media, and there's no extra biopsies to do and to manipulating with the cells. So therefore, there's less stringent requirements on equipment, on trainings, and on the techniques that you need to perform this assay. And also, it will significantly reduce the time for the lab to prepare and for the assaying the cell biopsies. And also, it will reduce the risk of the biopsy-associated complications for the later stage, and also it compatible to all the major NGS platforms. So This is the first clinical application of NICSI back to 2016. That the paper was published on the PNAS by Zhou, etc. And that introduced the first NICSI baby was born healthily, free of known genetic abnormalities, which could be inherited from the father. As shown in the table, the father is a carrier of a translocation, and on the left-hand side, we're showing the 3 embryos being tested by NICSI in the first instance, and subsequently 2 positive embryos were harvested and further analyzed to compare with the NICSI result, indicating the accuracy identification of these chromosome abnormalities inherited from the paternal origin, and also for the third sample, which is the actual baby being transplanted at the time as a healthy 46XY euploidy embryo to deliver this lovely and healthy baby, as shown on the screenshots of a local TV news report. And as NICSI and as also the non-invasive PGT is a very new techniques. So the researchers are debating how compatible or comparable its result to the T biopsies based PGT. In collaboration with Professor Catherine Rakowski in Harvard University, this research has been published on 2019, is to show that the NICSI assay for screening aneuploidy may be more reliable than the T result. And I'm going to show you the design of the whole experiment and the results. So this diagram shows the workflow of the experiment to compare the PGT-A analysis, use the T biopsies, the whole embryos, as well as the sperm culture media. So the whole embryo was used as gold standard for this study. The conventional T biopsy and the sample then sent to the test lab. And the result is being verified. The positive result is being verified by the third party lab and independent laboratory to confirm it's positive. And then these embryos, after the sampling of T, will be frozen down and the time. And the positive embryos will be was donated for and to be sold and cultured for another 24 hours. So subsequently, the culture media was collected for NICSI. At the same time, the whole embryo was also collected for the analysis. So this study setup enables us to directly compare the T result with the whole embryo result, as well as the NICSI result to the embryo. So this figure on the top shows the possible real-world scenario of the embryo mosaicism in the T biopsies for PGT-A and for the SEM when using NICSI. This is a false negative result of T is due to the cell being sampled, which are actually euploidy, while missing the aneuploidy T or ICM samples in the embryo, and vice versa when the sampling of TE being applied to, by chance, aneuploidy cells, then where other parts of the embryos are actually euploidy, then the first false positive result may be yielded. So the advantage of the using the SCM, so the sperm culture media, is that this is a collective genomic material of the whole embryos, so that will reflect both the ICM population as well as the T populations to contribute the DNAs, and that will allow a better estimations of mosaicism conditions of the whole embryos. And actually, it is indeed can be found in the table here, showing the Nixie has a much lower, so 20% versus 50% false positive rate, and a much higher specificity of 80% compared to 50% in TE biopsy. Therefore, we conclude that the Nixie, the PGT-A using Nixie is less prone to the errors associated with the mosaicism of the embryos, and would be more reliable than the TE-based PGT-A testing. So after Nixie has been developed, we have kept improving this technology based on the feedbacks from the clinician, from the clinical applications, and the data accumulated around it. So we have made certain changes and improvements for the following 4 sessions. So the sampling strategies, the whole genome amplification method, and the AI priority ranking systems were introduced for an improvement of the data analysis and reporting systems. So first of all is the sampling strategy. So we have introduced this the removing of this granular cumular cells on the day 3, even has been cleaned up during the ICSI procedures, and you do see the removal of the cells afterwards before there's several cells that could be the source of maternal genomic DNA contributions to the culture media has now been removed. And after removing this, then we change the media again to make sure that there's no carryovers of the maternal material for the downstream cultures. And this slide shows the sampling strategy that we have improved. For originally, we are sampling for day 3 to day 5, day 6 culturing medias, and now we are starting to performing day 4 to day 5, 6, or even day 5 onwards material for the downstream assays. And this is because that increased the concentration of the starting DNA, because there's obviously further development of the embryos will have increased number of cells, and so therefore more materials will be releasing into the culture media than the early stage. And also it can help to reduce the maternal contamination from the granular cells. And this is the reason why doing so. So we have compared the detection rates of different sampling methods. So from, you can see here, from day 3 to day 5, we have a concordancy rate recorded at 77.2%. We're comparing to, if we're switching to the current recommended day 4 to day 5, 6 sampling method, that can improve to 84%. And also you see the drop of the false positive rate from almost 15% to almost down to the half of 8.5%. And similarly, false negative result also dropping using this new approach, as well as the major concern of the false negative result contributed by maternal contamination can down to less than 5%. And the detection rate is remaining the same for all 3, but still for the day 4 to onwards sampling method, it allows to improve to almost 98%. And further down the line, we have a improvement of our library preparation and whole genome amplification strategies. So for the conventional, the first generation NIXI, that we perform the whole genome amplification, and then on top of that, do the library preparations. But now we have merged that two step into a single reactions within 2.5 hours. And after that, the library will be ready for sequencing. So it will be a huge amount of time savings. And before it was a manual curated data analysis, but now we have adopted for fully automated system. So the total time for this whole assay would be shortened to 9 hours, and that will be a significant jump from a 1 day or 1 day and a half procedures. And most importantly, that we have introduced the AI priority ranking for the embryo transfer for our NIXI. So we have collected over 300 samples of blastocysts that generates both NIXI results and whole embryo, so whole blastocyst cystic result to verify the real and true euploidy probabilities, and using that to give a grade of A, B, and C to our NIXI testing result. So the grade A is indicating a priority, the highest priority for the transplantation, and the probability of euploidy for grade A embryo would be over 94%, where the grade B sits in the between of C and A, where C is containing informed risk of chromosome abnormalities, and with the odds of euploidy down to less than 70%. So using these grading systems enable us to reporting a AI-based priority rankings to guide the clinicians for embryo transplantations, for embryo implantations. And finally, we have improved this automated analysis platform on ChromeGo. So it adapted to currently 2 major, but future will be 3 major sequencing platforms, as well as you can have a CM profile of the entire genome of this tested samples, as well as zoom into each individual chromosomes. So further down the lines, the study has been conducted, a retrospective studies has been conducted of this NIXI after being optimized using this AI-based training data set of that 300, over 300 embryos. So the implantation is first in this design of the study is based on its morphologies, but many times we collected the culture media and performing the NIXI. So after the clinical outcome being acquired, there's a clinical pregnancy of 101, and with subsequent ongoing pregnancy of 83. Then we use this data to look back, say, giving the NIXI grading of A, B, and C, what is the pregnancy rate, the miscarriage rate, and ongoing pregnancy rate. And in the next slide, we'll show that by reviewing this data, that is, there's 79 embryos were implanted based on the morphology are actually grade A, which is a fully euploidy embryos. And there's 40 of grade B and 41 of grade C embryos respectively. And you can see in this slides showing the pregnancy outcomes of these three groups. So the sign is A, the orange is indicating the group B, and the green bar indicating the group C. So you can see there's no significant statistical significances of the pregnancy rate, but you do see an increased trend of a successful pregnancy rate of 68.4% versus the around 60% of B and C grading embryos. But you do see a significant ongoing pregnancy rate in the group A embryos, which is 63% versus 50% on grade B and only 30% on the grade C. And also there's a significant reduce of the miscarriage. So the miscarriage rate is down to only 5% for the grade A embryos, where for the grade C embryos, which is not recommended for implantations, it is as high as 54.8%. And there's also another pilot clinical studies being performed by Fang, etc., which published on Journal of Translational Medicine on 2019. And this is also showing a very encouraging clinical pregnancy rate using the NICSI result for implantation as high as 58% and a low miscarriage rate of 10%. And in comparison to the internal data of single blastocyst transplantation versus the NICSI result, you do see a significant increased clinical pregnancy rate from 54% to 45.3% to 58%, and also a halved early miscarriage rate comparing to the SBT group, and also a much higher 52% versus 38% ongoing pregnancy rate. So indicating that NICSI does decrease miscarriage rate while improving the pregnancy rate. And after these single center pilot clinical trials, we're happy to announce that the multi-center clinical trial is also going well currently for the NICSI procedures. So sponsored by Peking University, third hospitals, and there's other collaborators around China to contribute. And basically this trial is to explore whether NICSI can be used as effective indicators for embryos selected either based on morphologies or compare using the NICS plus morphologies. And we were comparing the differences of live birth rate, the pregnancy rate, and the miscarriage rate between these two groups of embryos being selected. And this study will hopefully be completed by next year. So in conclusion, we have gone through the non-invasive capacity screening NICSI, which is one type of a leading non-invasive PGT techniques. And this NICSI techniques using AI for ranking the priorities of the embryos before implantation. So that would be a very helpful guide for the IVF centers. ICSI has also a great negative prediction rate. NICSI has also very great negative prediction rate of over 90%. So for the next stage, we will further develop this AI-based models with more data acquired from clinical studies and clinical trials. And also we have this multi-center RCT ongoing, basically using the NICS versus morphologies, hopefully to provide a better understanding of this new technology and what that can bring to us. So whether by then we will see whether it is real the time for us to moving into the era of non-invasive PGT. And thank you for your time.
Progesterone is a steroid comprising of 3 cyclohexane rings and 1 cyclopentane ring. It is manufactured for therapeutic use from the wild yam root Dioscorea species. Due to rapid first-pass liver metabolism, progesterone has variable concentrations and very low oral bioavailability. Therefore, it is administered via injection or vaginal route, which are not easy to use and associated with side effects. Dydrogesterone was introduced for clinical use under the trade name Dufaston in the 1960s for treatment of conditions associated with progesterone deficiency. The manufacturing process of dydrogesterone is highly complex. It starts with the chemical modification of progesterone, followed by a critical phase of transformation into dydrogesterone. This is done by passing it through UV light at an appropriate wavelength, intensity and time sequence, resulting in a unique, modified retrosteroid structure. This manufacturing process makes dydrogesterone the only commercially available retroprogesterone. The bent conformation of oral dydrogesterone enhances the rigidity of the molecule compared to progesterone. Due to its unique structure, dydrogesterone is highly selective for progesterone receptors, supplementing deficiency of endogenous progesterone. It has negligible interaction with other steroid receptors, resulting in a lack of androgenic, estrogenic, glucocorticoid and mineralocorticoid effects that can occur with other progestogens. Dufaston, containing 10 mg dydrogesterone in each tablet, is formulated to have good bioavailability, enabling convenient, patient-friendly oral administration. Improved oral bioavailability and the receptor agonist activity of its metabolites causes secretory transformation of the endometrium, progestational activity, at a dose 10 to 20 times lower than micronized progesterone. Various progestogens have been investigated to support endogenous progesterone in the treatment of luteal phase deficiency. Only Dufaston, progesterone and 17-alpha-hydroxyprogesterone caproate are currently approved for clinical use during pregnancy. In addition, Dufaston has a wide range of indications to supplement endogenous progesterone deficiency. It is shown to be effective in irregular menstrual cycles, dysmenorrhea, endometriosis, premenstrual syndrome, and secondary amenorrhea. In women undergoing assisted reproduction, Dufaston supplements endogenous progesterone and provides orally effective luteal phase support, contributing to successful and comparable pregnancy outcomes versus vaginal micronized progesterone. To date, around 113 million women throughout the world have experience with Dufaston, including 20 million fetuses exposed to dydrogesterone in utero.